The overall objective of the project Plasma-Based Treatment Processes for PFAS Investigation-Derived Waste was to investigate soil washing, followed by treatment of the soil washing solution in an enhanced contact plasma reactor to destroy desorbed PFAS. The IDW samples we used in our experiments were obtained from 4 sites, courtesy of GSI Environmental. Total PFAS concentrations in some of the IDW were up to several thousand micrograms per kilogram. Concentrations varied significantly between samples and also across compounds. Soil properties, including pH, anion exchange capacity, and organic carbon content also varied. The hypothesis we based our soil washing work on was that PFAS were sorbed to soil by a combination of hydrophobic sorption and ion exchange mechanisms. We therefore examined a combination of water, methanol, and salt to remove the PFAS from soil, similar to what has been shown to be successful for regenerable ion exchange resin. In our soil washing experiments, the addition of methanol increased removal. Statistically, across all of the compounds we analyzed for and across all of the IDWs, the highest removal occurred with a mixture of 50-50 methanol and water. We also investigated how salt content impacted removal using 50-50 dimethanol solution and found that 1% sodium chloride by weight was optimal. For the optimal wash solution, we found that 80 to 90% of PFAS was removed by the first extraction, 8 to 13% in the second extraction, and 0 to 3% in the third extraction. We also found that removal was fast, on the order of minutes, but this was for pulverized soil tumbled end over end, so mass transfer limitations were minimized. Now turning to the plasma treatment results. For these experiments, because we did not have much soil washing solution available, and in practice the methanol would likely be recovered through distillation, we used lab-prepared solutions and still bottoms from ion exchange regenerant solutions, courtesy of ECT2, to simulate soil washing solutions after methanol recovery. Plasmas are formed when the voltage applied between 2 electrodes is high enough to cause the gas in the space between the 2 electrodes to become a cloud of ions, electrons, and neutral species. In our reactors, plasma is formed using a high voltage discharge generated by charging a bank of capacitors and discharging it using a rotating spark gap. The discharge produces a very rich chemistry, including reductive species like aqueous electrons. Plasma discharge is unique compared to advanced oxidation processes in that it is both a reductive and oxidative technology. In our reactors, we concentrate the PFAS at the gas-water interface using bubbles produced by diffusers, so the PFAS will be near the discharge. To generate plasma, a high-voltage sawtooth electrode was placed above the water, and the diffuser was in the bottom of the reactor. The water volume was approximately 1.5 liters. We used approximately 400 watts in these experiments, equivalent to a small hairdryer, indicating that the plasma process is very energy efficient. In preliminary experiments containing only PFOA in a high brine solution, the starting concentration of 110 milligrams per liter was reduced to near detection limits, tens of nanograms per liter, in about 50 minutes. Shorter chain perfluoroalkyl acids were produced and then destroyed with an accumulation of PFBA at the end of the experiment. Significant amounts of fluoride were found in the water after treatment, generally accounting for up to 70% of the theoretical amount. The poorer removal of short-chain perfluoroalkyl acids is because they are not concentrated as readily at the gas-water interface by bubbling, as are the longer-chain compounds. In actual still-bottom samples, we had good removal of the longer-chain alkyl acids and slower removal of the short-chain alkyl acids. This slower removal was enhanced in these experiments by shorter chain alkyl acids produced by the breaking down of longer chain precursors. Precursors were removed during treatment and partially converted to perfluoroalkyl acids, which are then removed. Unidentified precursors were also present in high concentration in our samples. The top concentration was significantly reduced in these experiments after treatment. To address the removal of short-chain alkyl acids, we recently performed some experiments in which we added a cationic surfactant to a PFBS-containing solution. The surfactant associated with the PFBS and bubbling brought the PFBS surfactant combination to the surface where it was destroyed by the plasma. The surfactant CTAB, being a hydrocarbon, was also removed rapidly. We think this may be an efficient way to remove short-chain perfluoroalkyl acids if that is important for a particular site. Overall conclusions. Soil washing with methanol and sodium chloride is effective at removing PFAS from soil, although water alone is fairly effective, especially for short-chain PFAS in low organic carbon content soils. PFAS are removed from soil quickly, minutes in these experiments. Plasma treatment to destroy PFAS in high concentration salt solutions is effective. Precursor concentrations are an important determinant of required plasma treatment times. Precursor oxidation before plasma treatment would decrease treatment times and may be economically beneficial. Benefits to the Department of Defense. A significant amount of PFAS containing IDW will be generated in the coming years. An on-site desorption method combined with a destructive treatment technology would decrease IDW disposal costs and limit DoD future liability associated with PFAS going off-site. We'd like to thank project team members from Clarkson University, GSI Environmental, and ECT2. From Clarkson, Tom Holson, Selma Thagard-Mededovich, Michelle Crimmy, Justin Maselli, Sujam Fernando, and Raj Singh. From GSI Environmental, Steve Richardson and Poonam Kulkarni. And from ECT2, Steve Woodard and Mike Nicholson.